Good morning. It's December 18th, and this is your daily brief in science. Here's everything you need to know. Titan may not host a global ocean after all. A new analysis suggests its interior is dominated by deep ice, slush, and pockets of melted water rather than a moon-wide ocean. Using Titan's tidal lock with Saturn and gravitational interactions, researchers tracked surface and interior deformation, noting about a 15-hour lag between gravitational tug and the surface response to infer interior properties. While the model is compelling, some scientists, including Luciano Yes, caution that the evidence is not yet decisive, and Titan could still be an ocean world pending more data. The study is led by Flavio Petrica and Baptiste Journeau with collaborators from NASA JPL UW and international partners, and funded by NASA, the Swiss National Science Foundation, and the Italian Space Agency. Collaborators span NASA, UW, JPL, and international institutions, supported by funding from NASA, the Swiss NSF, and the Italian Space Agency. Future missions are expected to advance understanding. NASA's Europa Clipper will map Europa's ice shell by 2030, and ESA's JUICE will provide complementary data that could inform interpretations of icy moons like Titan. Cassini's long-running mission, from its 1997 launch to Titan flybys and the Huygens lander, provides the data heritage that informs the current reinterpretation. The NASA JPL-led study reanalyzes Cassini radio Doppler data with a novel processing approach to reduce noise and reveal deeper energy dissipation signals inside Titan. Published in Nature, the work ties energy loss and radio frequency measurements from Cassini flybys to interior properties and the viscosity of Titan's interior. Even with revised interior models for Titan and Europa, both moons remain high-priority targets for astrobiology, with ongoing analysis of existing data alongside future missions. Lead author Petrica and co-author Journeau acknowledge initial doubt and eventual support for the slushy interior model. NASA's Dragonfly mission, planned for the latter part of this decade, could help clarify Titan's interior and assess habitable environments, guiding future exploration. A new study backed by Nature shows a targeted approach to temporarily boost the aging immune system by turning the liver into a short-term factory for immune signals. MIT researchers used mRNA LNPs to deliver DLL1, FLT3L, and IL-7 to the liver of aged mice, creating a transient local boost that increases thymus-derived signals and raises T-cell numbers and function. The rejuvenation translates to stronger anti-tumor responses, with more CD8 T-cells observed in tumors and improved outcomes to immunotherapy, including complete tumor suppression in aggressive melanoma models in aged mice. The approach aims to counter immune aging by rebalancing multiple immune compartments through a time-limited intervention with no observed autoimmunity or liver toxicity. Findings were published in Nature on December 17th and were presented earlier at the American Society of Hematology meeting in Orlando, indicating cross-validation across venues. The Nature article highlights collaboration with Feng Zhang's Broad Institute lab and provides detailed methods and rationale for translating this approach to broader aging and immune contexts. The therapy relies on the liver's protein secretion capacity, with the signals produced short-lived to permit controlled dosing and reduce risk. All three factors, DLL1, FLT3, and IL-7, prove necessary for full immune enhancement, with loss of any one factor diminishing efficacy, indicating a synergistic effect. Off-treatment follow-up showed thymic output returning toward baseline while splenic T and dendritic cell numbers remain modestly elevated, suggesting transient yet durable gains without long-term autoimmune risk. The treatment required multiple injections over 4 weeks to sustain liver production of the factors due to the short-lived nature of mRNA. Researchers plan to test the approach in additional animal models, explore extra signaling factors to boost immunity further, and study effects on B cells and other immune cells, with ongoing support from HHMI, Broad Institute, and philanthropic sources. The study frames age-related immune decline around thymic involution and reduced Notch, Illinois, 7, and FLT3L signaling, which previously limited naive T-cell production and immune diversity. A new study in Proceedings of the Royal Society B finds Patagonian pumas increasingly preying on Magellan penguins along the Monte Leon National Park coast, signaling a shift in predator behavior tied to abundant prey. Researchers report penguin presence correlates with higher social tolerance among pumas, with those near penguin colonies about 3 times more likely to interact than elsewhere. Pumas that feed on penguins engage with each other far more often, recording 254 encounters versus 4 for non-penguin eating individuals, suggesting reduced competition for this food source. Monte Leon rangers supported the research, illustrating how conservation work can influence predator-prey dynamics and overall ecosystem structure. The findings urge cautious, nuanced expectations about carnivore restoration and its ecological benefits, highlighting complex ecosystem responses. The study also raises questions about broader effects on guanacos and penguin populations, which appear stable or increasing so far. Researchers emphasize the need to monitor Monte Leon A1S dynamics to understand potential long-term effects on pumas and penguin colonies. Penguin predation is surprising but plausible, with discussion extending to wider implications for wildlife restoration and management. Published in 2025, the work informs conservation planning by showing how trophic subsidies and ecosystem changes shape predator behavior and social structure. A polar bear family near Churchill, Manitoba, has surprised researchers by adopting a cub in spring and later showing a second untagged cub in fall, with the goal of identifying the cub's biological mother through genetic samples. Scientists, led by Evan Richardson from Environment and Climate Change Canada, tracked the family with GPS collars and field observations, noting the adoption of a cub designated X33991. The 2 cubs are about 10 to 11 months old and are expected to stay with their mother until about 2.5 years of age. The adopting mother is part of a long-running study that includes tagging and monitoring by Polar Bears International and partners from the University of Alberta and ECCC. The finding comes as polar bears are listed as vulnerable by the IUCN, with sea ice loss cited as a major threat to their survival. The broader context includes the western Hudson Bay subpopulation, which has declined roughly 30% since the 1980s, from about 1,200 to around 800 bears. Researchers describe the behavior as a positive example of maternal care that extends beyond direct kin and contributes to discussions about resilience amid climate-driven ice loss. The team notes the event as a meaningful observation of polar bear life history and care possibilities beyond biological ties. The long-term study tracks bears via GPS and makes some data publicly accessible, underscoring ongoing climate-related pressures on western Hudson Bay polar bears. The story emphasizes that, as the cubs learn hunting skills on the move to sea ice, maternal care remains a crucial factor in their development and potential independence. A plant-based plastic that dissolves in seawater while matching traditional plastics in strength and clarity could redefine how we think about pollution. Researchers led by Takuzo Ida at RIKENCEMS describe CMCSP, carboxymethylcellulose supramolecular polymer, as a material with full recyclability in a closed loop using electrolytes, and tunable flexibility from rigid to supple, even in very thin films. Importantly, it dissolves in seawater with no residues or microplastics, addressing marine pollution concerns. The work builds on a 2024 saltwater degradability advance and pushes toward real-world manufacturability, aiming for deployment beyond the lab. Core feedstock comes from abundant cellulose, emphasizing a plant-based foundation rather than petroleum. A safe, FDA-approved cross-linking system enables rapid dissolution in marine environments, while a protective surface coating prevents premature breakdown during handling. The researchers highlight real-world impact through inexpensive, biodegradable ingredients that enable recyclability and processability. The broader context notes prior saltwater melting recyclable plastics as part of ongoing efforts towards seawater dissolving materials and potential desalination implications. The 2025 study, published in the Journal of the American Chemical Society, reports a saltwater degradable plastic with 0 microplastics, derived from cellulose-based chemistry using carboxymethylcellulose and positively charged guanidinium ions. In summary, this material matches traditional plastics in performance, remains transparent, and offers dissolvability in seawater without contributing microplastics, signaling a potential path toward environmentally friendlier plastics on a practical manufacturing scale. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.